Now today, Professor Wendel is going to talk to us about genes and genomes, what we know about the evolution of Gossypium and the origin of the polyploids. And I was very fortunate last night to read a couple of your papers, Professor Wendel, and I can tell everyone it's going to be a really fascinating subject. So if you have any questions, please put them in the Q&A part of the chat. And with that, Professor Wendel, over to you.
Well, thank you very much, Kai, for that lovely, embarrassing introduction. I'm greatly flattered by it, and I'm honored to be speaking to all of you today. I really appreciate the invitation. It's always fun to talk about something that one has been passionate about for over 35 years. And so today I'd like to share with you a topic that I hope some of you are interested in. It certainly involves a plant that you're all interested in, cotton. And I'll start by sharing my screen. And hopefully, can someone tell me that you are seeing this? If you are, this is good. Is everybody seeing this?
We can see that, yes.
Wonderful. Well, you probably all know what you're looking at. And I don't need to tell you much about this plant. You all appreciate this, and you come at this from a number of perspectives. Many of you with economic or commercial or growing applications. Well, I'd like to talk about cotton today, but instead of talking about this cotton, I want to talk about this cotton. That is the genus Gossypium, the cotton genus. What do we know about it? How do we know what we know about it? And what does this have to do with the plant in the foreground that you're looking at? And it turns out that the genus Gossypium, which includes the cultivated cottons, has this extraordinary evolutionary history. So I'd like to talk about that evolutionary history today, in the process covering what we know about its taxonomy, how we know what we know, the evolutionary relationships among the species, tell you just to scratch the surface of this incredible, extraordinary evolutionary history that has been pieced together by many workers over many, many years. It's truly one involving multiple transoceanic voyages, chance meetings between species who have no business interacting with each other, but they do, and then they hybridize, and then new things pop out of that. Extraordinary genomic mergers. I talk about that. So I'm going to talk about the whole genus today. So cotton, which is important in the southern tier of the United States, is a relatively recent immigrant to the United States. It's only in the last several hundred years, following colonization of the New World by Europeans and then enslavement of Black Africans, that we were able to develop this into the important crop that it is today, from whence it spread worldwide. I am not in one of these areas. I am embedded in an ocean of corn and beans here in central Iowa. Just for perspective, that's where I am. And there's a little person holding dollar signs because my lab has been funded by Cotton Research for decades now. These modern annualized forms, here's an example for a Pima cotton, with this abundant flowers and fruits and long, strong, fine white fiber, have been derived by millennia of efforts by humans. Most of the progress made before DNA was discovered, in this case, by at least a 7,000-year history of domestication, starting with these photoperiod sensitive tropical antecedents you see on the left, bearing sparse flowers, a few here, a few there, with short, dark, tightly adherent lint. And this is the process of domestication. And it involves starting from wild forms, going through early cultigens, land races, and ultimately, just in the last few hundred years, developing annualized forms, and then ultimately the modern varieties that dominate cotton commerce today. One of the truly remarkable aspects of cotton domestication is that this happened not once, not twice, but 4 times. Twice in Africa, Asia, and twice in the Americas. The ones from Africa, Asia are diploids. The ones from the New World are allopolyploid. It's allopolyploid, and that means they contain two different genomes. And many of you know this, but I'll cover that. And this is what humans have wrought. This is 3 of the 4 domesticated species showing a single seed from the wild forms on the bottom and a domesticated version on the top. And these were all derived following thousands of years of human domestication, all transforming this single cell from this wild single-celled trichome that you see on the bottom to the domesticated version on the top. And wouldn't it be great if we understood how that happened? Wouldn't it be great if we understood the genetics and genomics of that? If we can understand how nature did this via strong directional selection by humans, then imagine what we could do if we had our hands on that understanding. Perhaps we could continue the process and develop much finer, better cotton today. The reason I don't have all 4 species here, I just have 3 of the 4 domesticated ones, is we've never found a wild ancestor for the 4th one, Gossypium arboreum. No wild ancestor has been discovered. And in fact, wild ancestors of all of these species are extremely rare. And this is one of my points that I want to drive home with a lot of examples today, is about germplasm resources and the importance of understanding them and the scarcity of these resources and the lack of global investment and concern about these resources today, because it's not a sexy topic like genomics and recombinant DNA and all that sort of stuff. But it's amazingly important, and everything we know most of what we know derives from work that was done long before Watson and Crick and DNA and all of that. Okay, so those are cultivated species, and in the upper right part of this slide, you see another example. I put this sheet on white paper so that you could see what humans have wrought from antecedents that look like plants on the upper left, but Gossypium as a genus has about 50 species. And seeds of the other species look like those in the lower left or those at the bottom. That's Gossypium davidsonii from Baja California. The lower right is Gossypium thurberi, one of the only 2 there's 3 really, American species. That one gets up into Arizona. That's Gossypium anomalum on the left from Africa. And you can see the fibers are much shorter. They're actually the same density, the same numbers in the cultivated forms. So the genus has about 50 species. Here's where they are. And you can scan your favorite parts of the world. You can see that they're largely between the Tropic of Cancer and the Tropic of Capricorn. They're largely shrubs that grow in very dry places. I've been to many of these places, not to all of them. Most of these species exist in small, isolated, relictual-like populations. Here's what one looks like. This is Gossypium australe from central Australia. You can see they are shrubs that grow in dry places. Fun to go there and collect. Look for the cotton in this picture, everybody. See if you can find it. We're in the Sonoran Desert, and many of you have focused right in on this plant right here, which is, of course, a cotton species. Now, a taxonomist, a specialist in the Malvaceae, might recognize this as a member of Gossypium, but most people would not. So that's cotton there. Okay, how about finding the cotton in this slide? Well, that's right. It's that big tree. That big tree in the middle is Gossypium aridum in full bloom. It's about 10 meters tall, maybe more. And you can see the little pink flowers tipping the branches. It flowers when it's nude, so to speak. It's lost all of its leaves. It flowers in the dry season. Here's what the flowers look like in an Iowa greenhouse. Spectacular flowers. You can see the extra floral nectaries at the bottom. Just extraordinary diversity in the genus. Let's go to Australia for a second. We'll go to Australia twice, maybe 3 times today. If you go in the upper left, you see the Kimberley region of Northwest Australia. There's 17 species of Gossypium up there. Well, we recently described a new species of cotton, and that's a story I'll return to several times. And there The Gossypiums are species that grow in these lateritic soils. It's in an environment where they get monsoon rains. They get 3 meters of rain in about 3 months, and then nothing for the rest of the year. And the vegetation burns. They're fire adapted. They send out these long vegetative runners. The capsules turn upside down at maturity, and they release onto the ground seeds you see on the lower left. Where's all the lint? It's very, very short. Evolution suppressed it because they've hired ants as mercenaries to protect and defend the plants. And in return, the plant provides the ant with a nice little dose of fat body. You see the fat body, it's called an elaiosome on the side of the seed. And the ants run off and carry the seed and plant it, as you see on the bottom right part of the screen. Extraordinary, marvelous evolutionary stories of coevolution with ants. When you go collecting here, you get lots of little formic acid injections as they're biting you, viciously defending the plants. So here's a slide courtesy of Cotton Incorporated, Ed and Don and anyone else who might be there from that august organization. I'm not sure if I contributed pictures to this slide, but there they are. Isn't that marvelous? This is the diversity in one genus of plants, the genus from which cultivated cotton is derived. It's extraordinary. Here's another picture of diversity in the cotton genus. And you'll recognize here some of the genome groups, perhaps some of you will. All those capital letters are genome groups. So you see, for example, in the lower right, you see F1, F is in Frank, that's B, that turns out to be a rare plant that is amazingly important. Why is this rare plant so important? Well, we know that it is the closest relative, the chimpanzee to the human, if you will, of wild, diploid, cultivated, which is now cultivated cotton, Gossypium arboreum and Gossypium herbaceum. So that transformation of the cell type, the original or the origin of long lint, if you will, is in a wild African plant. And we know its ancestor. And so now we're in a position to actually look for what makes those two different from each other. And then look on the left, what cultivation, how cultivation and domestication over thousands of years has improved those. So there's this wealth of genomic resources in Gossypium. There's these 50 species. How do we know that? Well, we are all standing on the shoulders of giants, as many of you know. And I wish to really give tribute to them today, because I'm talking about 300 years of plant explorers and taxonomists, highly trained taxonomists from many cultures, many different countries, focusing on the Malvaceae, the family to which Gossypium belongs, figuring out the patterns of variation in nature, what might constitute a species, how it might be related to another species, passing down that knowledge over generations using the scientific method, the best tools of the day. And so you might recognize some of their names like Rafinesque or Parlatori, or you might have written out, say, Gossypium thurberi, and after that name seen Todaro. Todaro, the scientific name actually has the generic name, the specific epithet, and then the author. Those are the three components of any scientific name. Todaro, it was the original application of the name Gossypium thurberi. And he wrote a book summarizing what we knew about cotton at the time. And then Sir George Watt wrote another very important book called The Wild and Cultivated Plants of the World. In fact, you'll see Watt's name if you write out Gossypium darwinii from the Galapagos Islands. Watt was the author of that. Anyway, he wrote a revision of the genus Gossypium. Excuse me, terrible frog in my throat. Well, that would be 113 years ago. I have this book. If you look at the subtitle, it says, framed primarily with the object of aiding planters and investigators who may contemplate the systematic improvement of the cotton staple. So a lot of this taxonomic history was motivated by this economic objective. I really like this book. It's good reading. In chapter 1, he writes, it would not be far from correct to describe cotton as the central feature of the world's modern commerce. And I like that. It makes me feel good about that I'm working on something that might actually matter to the planet. And that's a nice feeling. So all these taxonomists were telling us about the wild species, but what about the cultivated species? Many of you know, and I've already told you, that there's 4 domesticated species. That wasn't always clear. There's tremendous variation in these species. As anybody who's been to a grow out of cotton germplasm from Barbadense and Hirsutum or Arboreum and Herbaceum, sometimes you're not quite sure what species you're looking at. They have a long history of introgression. They have a long history of gene flow, hybridization. Humans have been transporting them for thousands of years, where they've been meeting other germ pools, gene pools, and interbreeding, and so it's a mess. There are over 250 taxonomic names that have been applied at one time or another to a cultivated cotton. Well, it wasn't until about 100 years ago that this Russian, who died very young, made sense of all this, who decided there's really only 4 gene pools, 2 in the old world, 2 in the new. This was his great stroke of insight. It turns out to be true. This is validated by further great minds like Sir Joseph B. Hutchinson, who wrote many other important papers on the domesticated cottons. But Hutchinson, along with Silo and then my academic grandfather, Stanley G. Stevens, published a great book in 1947. It's about time for it to be redone, I think, called The Evolution of Gossypium and the Differentiation of the Cultivated Cottons. Hutchinson also told us about all these land races that are really, human constructs. They're not firm biological entities, but trying to encapsulate some of this tremendous diversity that exists in Gossypium. And then along comes Stanley G. Stevens. This is a picture at North Carolina State University, where I started as a postdoc in 1983, and those greenhouses in the background is where I first was growing my cotton, just because I was working on maize, but I was interested in cotton. S.G. Stevens was just a brilliant man. He was really a pioneer in our understanding of the importance of gene duplication and evolution, our understanding of hybridization and reproductive barriers and introgression. He wrote many important papers, the potentiality for long-range oceanic dispersal of cotton seeds. Wonderful paper, important. I'll talk about its implications later today. Stevens is the only member of the United States Academy of Sciences, the only one who specialized in cotton. And so they have a memoir series, a specific memoir series. So I wrote a biography of Stevens with Major Goodman almost a decade ago now. So if you're interested in Stanley G. Stevens, I refer you to that reference at the bottom. So that's a brief introduction to this diversity at the morphological level. Well, paralleling these couple hundred years of taxonomic investigation, about 100 years ago, we started having decent techniques for looking at chromosomes. And so a number of workers, shown here in the upper left, started working on cotton chromosomes, understanding the cytological basis of some of this diversity, differences in chromosome size, differences in chromosome number. And then Skovstad, at a very young age, made the algebraic insight that the number 26 is related to the number 52 by a factor of 2. And he actually proposed that the New World allopolyploid cottons contain 2 genomes, one from the Old World, one from the New. And then Beasley came along, and then later Harlan independently verified it. And they experimentally synthesized a Thurberi arboreum hybrid, colchicine doubled it, showed that it was fertile with hirsutum and that the chromosome pairing was as expected, and proved that the New World tetraploid cottons have Old World cotton. And so this led to a lot of mysteries. Who were the parents of the polyploids? How did this happen? When did this happen? So in the 1970s, I became interested in this classic botanical mystery. And then finally, as my final reference to someone who should be greatly honored by anyone who's interested in cotton, is this man, Paul Fryxell, who was the modern taxonomist, the expert in Gossypium. And Paul unfortunately passed away some time ago. He actually got his PhD at Iowa State University and lived on the same street where my house was for 30 years. And just a remarkable coincidence. But I encourage you to look up the natural history of the cotton tribe. It's out of date in terms of its taxonomy now because it was published in 1979. We've described new species since then. But it's marvelous with its understanding of ecology and evolution, the geography, the traits of different wild species of cotton plants. So as I said, and to paraphrase Sir Isaac Newton, shown here on the left, we are indeed dwarfs on the shoulders of giants. And these are all the people, the many others who I don't have time to go into, who carry us high and raise us up by their giant size so we can understand something about the genus Gossypium and its diversity. And then people like me can come along in modern times. People like me can come along because I happen to be born in an era, a couple generations after DNA was discovered, and then living in this breathtaking era, this genomic technology era in which we find ourselves, this golden era of biology. And so I can run a lab and start using this wonderful bag of tricks, these molecular tools, genome sequencing, to figure out how all these species are related to each other and when everything happened. And so we can figure out the phylogeny of the Gossypium. And it looks like this. This is in yellow is the phylogenetic history of the genus, and the little blue call out dots are different groups of diploid species. So I can talk about the D genome diploids from the New World, or the A genome diploids from Africa, you can see there, or you can see there's 3 different groups of Australian cottons. Well, this is nice. So we figure out the phylogenetic history using molecular tools. And we can use the molecular clock to figure how old the genus is. So we know the genus originated 5 or 10 million years ago. That's nice. So we have a time framework for everything. Now we know that all of these genomes all these diploid genomes have the same haploid complement of chromosomes, n equals 13. There's 13 chromosomes in each of these approximately 50 wild species. But their genomes vary tremendously in size. And now representatives of all these groups, actually today, I can tell you, have now been sequenced in full with high quality de novo assemblies. And even though they all have the same chromosome number, they vary threefold in genome size, from a low of 900 megabases in the American D genome group. That's a primarily Mexican clade of somewhere between 15 and 17 species. It's still a little unclear, all the way to 3 times that amount, 2,600 megabases in the Australian K genome clade, which is 17 species minimally there. Tremendous genome size, shrinkage, and expansion during the evolution of cotton. We can also superimpose morphological innovations onto a phylogeny. So there, if you look at Africa, you can see F is the closest sister group to A, and that is where nature first invented long trichomes, long single-celled epidermal trichomes that many of you call fibers. And then we can diagnose, because the genus is only 5 or 10 million years old, the continents haven't moved very much. So that means cotton has a fondness for oceanic travel. There must have been a colonization of the New World with an African origin. There must have been a colonization of Australia. Well, we know there's a wild species in Hawaii, a wild polyploid I get to go collect in Hawaii, usually during the wintertime in Iowa. Hawaii is the most isolated oceanic archipelago, and it has a flora and fauna, and somehow cotton got out there. There's 2 different colonizations of the Galapagos Islands. There's one, a very important one, of a rare species, Gossypium raymondii, in Peru. And this is the closest donor, the closest model of the donor to the origin of the allopolyploid cottons. And then there's this. It's so important, I'll show it to you again. Sometime in the mid-Pleistocene, 1 to 2 million years ago, an A-genome propagule managed to cross either the Atlantic or the Pacific, where it encountered a D-genome plant. How it made this voyage, we don't know. Cotton seeds sink. Most of them sink like rocks after a little while. After they get wet, they sink. But they're saltwater tolerant and they get around. It's the most amazing thing, cotton gets around. And so an A genome, now remember, longlint originated in Africa. It crossed one of the oceans, hybridized with a native D genome species 1 million years ago, perhaps, and gave rise to allopolyploid cotton. So cotton really gets around. It's just amazing. Well, there's a new record for long distance dispersal, and that's because the Chang'e 4 lunar module from China landed on the moon in 2019, robotically did a tissue culture experiment, and the very first plant ever grown on the moon turns out to be cotton. So I think cotton will hold the record for long distance dispersal for a very, very long time, certainly in our lifetimes. Okay, so back to this story of the origin of the New World allotetraploid cottons. You can see they combine two genomes. They're in blue, an A genome from Africa, a D genome from the New World. They're in the process, doubled the number of chromosomes, and then they evolved into the 5 species in nature that you see down below. And then later, humans evolved. And then later, much later, in the last 8,000 years, 2 of those species were domesticated by humans, Gossypium barbadense, which you might know as Pima cotton, and Gossypium hirsutum, or upland cotton. So that's the big story of the origin of cultivated cottons. What do those wild allopolyploids look like? Well, here they are. In the upper left, you have Gossypium tomentosum with its beautiful sulfur yellow flowers. This is an endemic to Hawaii. It's really interesting, kind of isolated species. You have in the upper right, Gossypium mustelinum from northeast Brazil. Only about a dozen populations known. It turns out to be the basal branch within the allopolyploid clade. In the lower left, you have a wonderful picture of the Gossypium from the Galapagos Islands, which has the lovely commemorative name, Gossypium darwinii, commemorating the island's most famous visitor, of course, Charles Darwin. And then in the lower right, you have what Gossypium hirsutum, upland cotton, looks like in nature. These are scattered individuals in these wide-ranging, these sprawling shrubs with sparse, small flowers growing fully integrated in native vegetation, often right down in the salt spray zone. They must harbor a lot of genes for drought and salt tolerance. And these forms first attracted human attention probably around 5,000–6,000 years ago in the north coast of the Yucatan Peninsula. They ultimately were developed, domestication traveled down the Yucatan Peninsula into Mexico, Guatemala, then northward and into the most of the gene pool that you recognize today as upland cotton. But if you go there today, this is from Colima in Mexico, you see plants like this. These are dooryard or commensal forms, and they're maintained locally for a diversity of uses, pillow stuffings, lamp wicks, things like that, hygiene products, and then land races, and then ultimately, just in the last couple hundred years, these modern annualized forms shown here. This is one from outside of College Station, Texas. And then we're spread worldwide. You can see here, this is a picture I stole from a men's clothing catalog. And this gentleman is holding Gossypium hirsutum, and it says cotton, one of Egypt's greatest treasures. Well, it's not native to Egypt. This is a post-colonial introduction where cotton spread worldwide. And what I like so much about this slide is if you look in the background, you'll see we are no longer in the New World. And it's right about the same time those buildings in the background were being built that this plant was being domesticated half a world away. And here, ultimately giving rise to the world's most important fiber plant, is shown in this picture from Jan Arthus Bertrand's book, The Earth from Above, showing a worker in the Ivory Coast resting on his work product. Okay, so we've come along with our bag of tricks. We've sequenced 700 genomes representing this full range of wild to domesticated. And I'm happy to tell you, you see here, Daojun Wan is first author in my lab. And the the other first author. This is a collaboration between Josh Udall and myself. We had an NSF grant to do this. And I'm happy to tell you that 3 hours ago, this was accepted for publication in Advanced Science. And so this shows you the gene pool of domesticated cottons, how it's related to all these landrace and wild forms. And so if you want to look at this paper, it'll be out soon. It'll be up on bioarchives any day. And it'll describe what I've told you about, which is the original domestication in the Yucatan Peninsula of Mexico, shown there in the middle of this slide. Its diffusion south and then ultimately north to develop the modern annualized forms. At the same time, spreading into the Caribbean, where it underwent a great deal of hybridization with Gossypium barbadense or Pima cotton, and then from there spread worldwide. And so we now know about the gene pool of cultivated cotton, and we can understand how narrow it really is. Cultivars, each little dot represents a different cultivar where we fully sequence the genomes, and you can see the narrow blob of green points relative to the diversity as a whole in this two-dimensional plot of diversity of about 700 Gossypium hirsutum cultivars. Meanwhile, in South America, about 8,000 years ago, Aboriginal peoples were attracted to this plant that had this fuzz that they could use for various uses. They brought it into domestication, as I show you here, ultimately deriving Pima cotton, the other really important allotetraploid commercial cotton. There's a lot of archaeological evidence of this, including 6,000-year-old blue jeans. They were dyed with you can see there a colorized image on the left. The natives used a member of the legume genus Indigofera, which you might know. The citation is there at the bottom. And this is the history of Pima cotton. So in the same paper, this is a very big paper that I described a minute ago. You can read about the origin and spread and diversification of Gossypium barbadense, which originated in Northwest South America and then spread both north and east of the Andes, and then from there worldwide to give you Pima cotton. So this is, again, continuing the story of where all of our cotton comes from. But I'd like us to reflect a little bit on taxonomy, diversity, the importance of understanding what nature gave us, not what we are creating. And so here I've shown you the non-algebra, 1 + 1 does not equal 2. If you take the A genome on the left and the D genome on the right, these are the best models of the species that gave rise to the plant in the middle, which is allopolyploid cotton. And this one right here is called Gossypium raymondii. It was almost extinct. We almost never described it. What would that mean if we had never found it? What would we think about the origin of Gossypium, of all the allopolyploid cottons? Here's what it looks like. There's a flower in the Iowa greenhouse. Below you show the A and D combination that gave rise to the allopolyploids. This species wasn't even described until 1932 by Oscar Ulbrich. So if you've seen Gossypium G. raymondii Ulbrich, that's this guy. Then a lot of people showed that this is among the best models of the D genome ancestor. We've sequenced it. It is the best model of the D genome of the allopolyploid cottons. Phillips states that the species was represented by relatively few individuals, and these are confined to a narrow geographic range. This is the main story. This is the rule rather than the exception for wild species of cotton. And then, sadly, there was a lot of efforts by the Peruvian government with aid from American chemical companies to eradicate it because there was some concern it could serve as an alternate host for the boll weevil. And so during the 50s and 60s, it was almost made extinct. It has not been collected in 35 years. This is where it's from, in a couple of departments in northern Peru. In the right there in the foreground, you see what Gossypium raymondii looks like. I think there's another expedition that's needed to go collect it. It's really in just a couple of river valleys. It's in these small scattered populations. And I've studied about 30 of these populations, and there's almost no diversity in it. So people need to go out into nature and collect and describe and figure out what's out there still. We're still describing new species. And we're losing the people who had expertise in this. Here's two of them. Many of you knew Max Stewart on the left. He passed away a number of years ago. So did Ed Percival on the right. These were the most joyous, skillful collectors of plants from around the world, giving us a lot of the resources that exist in our germplasm banks today that are very poorly funded, very underfunded. And there's a great risk. I'm like the last person standing who does this stuff in the US. I mean, there's one person who works for the germplasm bank in College Station, Texas, and how much can one person do? So bringing this sort of taxonomic perspective and diversity perspective to the genus, it's kind of a dying art, and I'm a little concerned about who's going to carry on this tradition into the future. And there's reasons to do it. If I take you back to Australia, we had a helicopter to go collecting. You can see the helicopter in the lower left. And I mentioned that this is the species here, fire adapted. Well, so everything in the vegetation is turning brown, except in the helicopter, I saw something glistening in the sun. And I said, what's that? Land down there. And we landed down there, and I knew within a millisecond, well, that's an exaggeration. I knew within 10 seconds I was looking at a new species of Gossypium. And here it is. That's me as a much younger man, because these gigantic petals were reflecting the sunlight, and I could spot them from the helicopter. And this is what the flowers look like, these fantastic mauve flowers. And we just described this as Gossypium aneuploides in the somewhat obscure journal Novon. That's a journal that publishes new scientific names. So there's new species of cotton still to describe. Okay, I'm almost done. I showed you this slide once before, and it said 5 species. Why does it say 6 now? Because I want to continue to make the point about new diversity that's still out there. So I've added the species on the right, Gossypium ekmanianum, to this slide. What is Gossypium ekmanianum? Well, it was described in 2008 in this very obscure journal, Bonplandia. And I said, what? And so here it is. I got the specimen and I looked into it, and it was first proposed by as a species by Whitmock in 1928. It's endemic to the right half of the island of Hispaniola, the Dominican Republic. It looks pretty wild. It has a tortured taxonomic history. It was reclassified as part of Latifolium by Robotic in 1942, reclassified as part of Hirsudium 8 years later by the same man, reclassified as Gossypium tricuspidatum by Maurer in his book in 1954. And then Paul Fryxell and other modern workers treated it as just a form, a variant, one of these sort of feral or landrace forms of Gossypium hirsudum. Well, there's this other really obscure publication, Cotton Collecting on Caribbean Islands in South Florida, again hearkening to the importance of collecting. And this is one of Jacques Schwendemann's trip with Ed Percival shown there in the middle. And in that little paper, I had underlined and stuck in my file cabinet an actual paper copy, not a PDF, that said that bourdon in an electrophoretic survey of this material showed that there was a lot of enzyme variability. Well, in the late 80s, I did allozyme analysis, if any of you know or remember allozymes, of about 500 accessions of hirsudum. So I dug into my files, and I actually had paper copies. And you see on the left, Dominican Republic. So I went and looked at these accessions, and I circle here for you in green some numbers that are not found anywhere else. And these are alleles that are completely novel, otherwise not found anywhere in 500 accessions of Gossypium hirsudum. So we studied it, published it in 2015 down below, that we confirmed that this is indeed a new species of allotetraploid cotton, Gossypium ekmanianum. All the DNA stuff you see there shows that AD6, Gossypium ekmanianum, the second row from the bottom, has this chunk of DNA inserted into it that nothing else has in the chloroplast genome. And so it's sort of diagnostic DNA feature of the novelty of this species. Okay, and now continuing our voyage around the world, let me take you to that red dot. Whoa, you are out there in the middle of nowhere now. You are in the Wake Island. What's the Wake Island? It is a tiny little spit of coral, about 3 centimeters. I mean, 3 centimeters, 3 kilometers across. And Ray Fosberg, who I knew was a Malvaceae specialist, was commissioned to write up the species list of what grew there. And he did that, and he described Gossypium religiosum, which is one of the many, many taxonomic synonyms of Gossypium hirsutum. But he said in that paper, the Wake Island cotton does not closely resemble either the Caribbean or other Pacific forms. So I thought, well, that's curious. Maybe I should look at it. So I talked to Paul Frixell, and this is in the previous millennium. And I actually had in my files a printout of an old email. This is shortly after we all started getting email in the 1990s, if you remember. And Paul stated that a seed sample of Wake Island cotton is on its way to me. And he got it from Daryl. Ray Fosberg gave it to Daryl Herbst. Daryl Herbst at the Bishop Museum in Honolulu. And Daryl gave it to Paul. Paul gave it to me. Daryl calls it hirsutum and says it may be akin to the Gossypium hirsutum from Tahiti. But my recollection from seeing the herbarium sheet was that it might show some kinship with Gossypium tomentosum. That's the wild Hawaiian endemic. So I was intrigued. So of course we got it. We started looking at it. We started studying it. There you are in the Wake Island. It's, you know, in the South Pacific Ocean. I mean, here you are. You are surrounded by thousands of miles of open ocean. And you see the long airway strip there. This is now run by the American military. It was important in World War II. The military established a presence there. I'm not allowed to go collecting there. But I did contact the military, and after about 50 letters and emails, it turns out that this is under the auspices of a branch of the Air Force that's in Alaska, if you can believe it. Well, they agreed to collaborate with me on this project. And so they went out there and they collected samples from the Wake Atoll here, which is actually 3 little islands. You can see there's on the left branch and on the right branch, there's 2 little islands and then a bigger island. And again, that's only 3 or 4 kilometers across. This is, you know, this won't be there for a little more ocean, a little more global warming. And it is indeed a new species of Gossypium. And I named it, when did we publish this? This is now, it's a little over 3 years ago. We named it, gave it the commemorative name, Gossypium stevensii. How do we know it's a different species? Well, we've now sequenced the genome, but in an early study, and this is what you find in the paper, if you can sequence 600 genes from a whole battery, all the other allopolyploids, that's what the little circle here on the left, that's Gossypium barbadensine darwinii, that group of allopolyploid cottons. Over here on the right, you have Gossypium hirsutum and its close relatives, Gossypium ekmanianum. In the middle there, in orange and blue, these are the more isolated species, Gossypium mustelinum, which is the first branch of polyploid evolution, Gossypium tomentosum from Hawaii. None of this variation includes the Wake Island cotton. There it is at the top. It's clearly a distinct species. We have no idea what traits it harbors. I'm trying to increase seed. It doesn't want to set a lot of fruits, but I'm trying to save it for posterity, the world. So there are still new species out there, and we gave it the commemorative name, Gossypium stevensii, not just because he was my academic grandfather, and not just because he was a brilliant man, but because Stevens was very interested in the potentiality of oceanic transport of Gossypium. So part of this remarkable history is this recurrent, mysterious travel on the open seas, sometimes forming new species. And there's Stanley G. Stevens one more time. Okay, that's my talk. On this one very busy slide is everything I told you, with the exception of the perspective that taxonomy matters, germplasm matters, diversity matters, our germplasm banks are critically important. It's difficult work to grow and maintain these wild genetic resources. It's not like growing petunia in your greenhouse where it's very easy to grow. It takes some tender loving care to get these things to flower and set seed and increase seed. And they don't necessarily last forever. Some of them have great longevity, some don't. But at the very bottom of this slide, on the left, you see A and D. Those are the 2 diploid donors. We've now been able to piece this together because my lab and my colleagues around the world are standing on the shoulders of giants, as I mentioned. And about 5 million to 10 million years ago, they last shared a common ancestor. They then diverged, and then sometime in the mid-Pleistocene, I wrote here 1 to 1.6 million years ago, they met each other again, having evolved a twofold difference in genome size. And despite that, they could hybridize. They could double their chromosomes. They gave rise to an entire clade of allopolyploid cottons, which then evolved into the now 7 species that I show there with names, from Gossypium mustelinum on the left, AD4, all the way to the newest one, Gossypium stevensii on the right, AD7. And this is all superimposed on a map of the world, showing in colors where each of these species is found, emphasizing again this magnificent attraction, fondness that cotton has for transoceanic, for cruising. Cotton likes to go on cruises. And that's it. If I left you with any questions, I'll be happy to answer them. We are the beneficiaries of generations of taxonomists, cytogeneticists, and plant collectors, to whom we owe a great debt of gratitude. They taught us that there's a genus out there called Gossypium, and we now know that it contains more than 50 species. Over the last 5 to 10 million years, it's achieved a global distribution, has a remarkable history of long-distance oceanic travel, and of polyploidy. And I've tried to emphasize the importance of biodiversity, of germplasm banks, and how thin this enterprise is in terms of our devotion of research resources to it. I'm 66 years old. I'm probably going to retire in the next X number of years, and I'm concerned about who's going to carry on this kind of work. Much remains to be learned, studied, preserved, and utilized. And then, obviously, I am standing on the shoulders of everybody in my lab and all my collaborators around the world, past and present, and am indebted to the many funding sources who have enabled any of this work to proceed, including the National Science Foundation, the USDA, and Cotton Incorporated. So that's it. I will stop my share. And then if any of you have questions, I encourage you to put them into the chat. I can open the chat and do my very best to try to address any questions you might have. Thank you again for the invitation, and thank you for your attention.
Well, wow. Thank you, Jonathan, for that really fascinating presentation. I certainly learned a hell of a lot today, and I know that everyone else now knows everything about allopolyploids. I've spent 50 minutes rehearsing that now, so I've got it right.
Excellent, excellent. You do indeed.
So there are some questions coming in on the chat, and I've got Dr. Cranthi who's going to help me with any other big words. So let's start with the first one, which is from PJ Kulkarni. How can diploid cotton species be used to improve pteroploids practically, especially Gossypium hirsutum?
Yeah, that's a great question. There's a recent review by Peng Chi, C-H-E-E, on the utilization of wild species for cotton improvement. I don't know if that, I don't remember the year. That might be 2017. But if you look up Peng Chi, P-E-N-G is his first name, C-H-E-E. I'm a minor author on that paper. Andy Patterson is an author on that paper. That might help you narrow it down. We wrote a review on how wild cotton can be used in improving commercial cotton. And now there's two main approaches. The classic one is through what's called hexaploid bridging, where you can take diploids, cross them times tetraploids, you make a triploid, you double it, you have a hexaploid. That can be fertile, and then you can get some introgression from the donor genome into the recipient genome, the recipient being hirsutum, or barbadense for that matter. The other way, of course, is nowadays transgenic work. It's still this technically transgenic work in cotton is still difficult, much more difficult than in many other species, but progress is being made, really impressive progress in China, perhaps in some other places is being made on doing transgenic work using CRISPR and Cas9 and the other developing transgenic tools for improving cotton without having to go through that whole introgression breeding program. But those are the two. That's a kind of a long answer to your question. I hope it addressed it.
Great. And we have a couple from India. First one is from Daminda Patak. Why do cotton tetraploid species behave as diploids cytologically?
That's a great question, Dharminder, and hello. I can't see you on my screen, but wonderful to hear from you. Dharminder is a great scientist, and I was fortunate enough to have him in my lab for a year, a long time ago. So we don't know. So in the process, many of you might know that when you cross diploids and make an allopolyploid, you often don't get bivalence. You get a mixture of multivalence at meiosis and messy cytogenetics. And that is indeed the case when you resynthesize tetraploid cottons. You get all kinds of cytogenetic abnormalities. But if you look at natural tetraploid cotton, you don't. They form nothing but perfect bivalence all the time. Nobody ever sees anything but bivalence. So that means evolution has acted with very strong selection to enforce bivalent pairing instead of multivalent pairing. And this happens in different ways in different plant groups. And there's probably on the order of 100 different proteins that have been implicated in enforcing this in experimental systems from Arabidopsis and from wheat. They're the best developed. But we think nature has found a lot of solutions to this, and we don't know the answer for cotton. It is a complete mystery. There isn't even a hint of how it might work yet in cotton.
Thank you. And from Dr. Santosh, also from India, is there any Gossypium species that has a determinate growth habit? And then his second question is, which tetraploid wild species contribute significantly to cultivated cotton improvement?
Well, let's take them one at a time. The first one, is there any cotton that has a determinate growth habit? Not a program determinate growth, but environmentally constrained determinate growth, most of them, because of their seasonality. Some are more perennial than others in their growth, but there's no, that I'm aware of, wild cotton species. I'm scanning all 50 species in my head while I'm answering, that has a strictly determined growth habit. No, you will always, you can stump all of them and have them keep growing from axillary branches. Yeah. And the second question was, I'm sorry, what was the second question again about tetraploid cotton?
It's which tetraploid wild species may contribute significantly to cultivated cotton improvement?
Potentially all of them. What we know is that Barbadense and Hirsutum have. That's where they sprang from. Those wild species gave us the two cultivated species that we know of as Upland and Pima. Pima is not pure Barbadense. Pima has plenty of Gossypium hirsutum in it. Because of this thousands of years, if you think about it, after original domestication, 5, 6, 7, 8,000 years ago, humans weren't sitting still. There's lots of histories of human migration. And so many different peoples, waves of immigration into the broad areas of the Caribbean, for example, brought these species with them, and they intermingled. So there's been multiple rounds of mixing in the gene pools over history. And that gave us all of our cultivated cotton. What traits do the other wild species have that we might be interested in? We don't know. I'd like to single out the fact that wild tetraploids, unlike wild diploids, wild tetraploids tend to be specialists in coastal zones. If you think about Gossypium tomentosum, I've collected it in Hawaii, growing on the north shore of Oahu in the salt spray. Think about Gossypium stevensii from the Wake Island atoll in the middle of the ocean. So I suspect that there's a lot of salt tolerance traits, or they're not like single genes, the salt tolerance physiologies that have evolved, that if we could understand more about how to manipulate them, those might be useful and perhaps introgressible from these wild tetraploids. They are all interfertile. It is possible to make crosses in every possible direction between wild cottons.
Thank you, Jonathan. Looking down, there's quite a lot of questions on a similar theme. One here from Bruno Bachelier from CIRAD. Do we know which potentially interesting traits, resistance to drought, salt, pests, diseases, etc., are present in wild species and could be used to improving cultivated species?
No.
Okay.
We do not know. And how could we know? Because these wild species have not yet been examined for most traits. They're too poorly understood, too poorly studied.
There's a comment here from someone who is perhaps saying that it may be possible that birds may have eaten the Gossypium seeds and dispersed them from one continent to the other. Question mark. So that's, do you reckon?
Yeah,
that's
I love that. I love that question. Yes, so that goes along with my favorite joke about cotton dispersal, which, no one knows how it made it the voyage across the oceans because the seeds sink. So I've always said it was a constipated duck blown off course in a storm that managed to cross one of the oceans. It is possible that birds will collect some cotton seeds. And in fact, I'm just so delighted to be able to share this with you. I actually have a collection, one accession of Gossypium darwinii from one of the islands in the Galapagos that was collected from a finch nest, Darwin's finches. So birds will collect them. Not many birds can make it across the ocean with a cotton seed in its gullet. And, I suspect that a lot of this is happening by perhaps mature but non-dehisced capsules floating on some natural, getting blown off coast in a gigantic storm and then floating on natural rafting. You might all know stories of tankers who sank in the North Pacific, but they were, carrying 100,000 Nike shoes or rubber duckies. And then people all over the world would follow these and collect them down the Pacific coast of North America or out into Hawaii again. Stuff does get around. It just takes a long time.
Great. And we are running out of time. So, Keshav, is there anything in any of those questions that you can see that would be really important that I may have missed? I think we've covered the main themes.
Yeah, thank you, Kai. And I can just see a couple of them. But before I ask these questions, I must say, Professor Wendel, your presentation was like a Hollywood thriller. It was so exciting. I enjoyed it thoroughly, and I wished you went on and on and on. But thank you so much.
Thank you for the honor.
Thank you. It's our pleasure. It's our privilege. Like this question from Professor Alabaksh from Turkey. He's asking, what are the interesting traits in newly discovered species that can be integrated into Gossypium hirsutum?
Oh, I wish I knew. I would point out, I mentioned it before, but I'll say it again. Gossypium stevensii is from the middle of the ocean. So for sure, that plant has figured out a way to survive both drought and salt.
Yeah,
writ large, right?
Yeah. And then there is a related question from Venkatesh from Raichur. He's asking, which wild species can be used to introgress heat tolerance trait into our modern tetraploid cultivars?
That's a great question. I'm thinking about all the wild species. We have a lot of wild species that grow in very hot areas. For example, there's some in Baja California, or in the Sonoran Desert. Or I'm thinking of the arborescent cottons that grow on the western slopes of the Sierra in Mexico, where it's hot as the dickens for much of the year, incredibly hot. And they've decided it's so hot, I don't even want leaves around. They drop all their leaves and they still flower. Those gigantic pink flowers I showed you on the tips of the 10-meter trees. Yeah, I'm thinking of plants from the middle of the Australian continent, where it's hot as blazes, one of the hottest places on Earth. And Gossypium australis, Gossypium nelsonii, Gossypium bickii are perfectly happy there.
Okay. Dr. Gerard Prasad from India is asking this question: is Gossypium arboreum evolving? His question is short. He says, arboreum evolution, question mark. Is it still evolving?
Everything is still evolving, including Gossypium arboreum. We haven't found the wild form of it yet, which is super interesting. It may be from the wild forms of it. We don't know if they actually originated in Africa. It could be the Gossypium arboreum originated farther east. Perhaps as far east as somewhere in the Indian subcontinent. We don't know. Paul Fryxell once was talking about a specimen from the Philippines that was lost in World War II in one of the bombings of the herbaria that he thought might be a wild Gossypium arboreum. But it's never been found, the wild ancestor of arboreum. One of the proposals was that arboreum actually was not a wild plant, that it evolved from herbaceum under cultivation. But we've put that idea to rest with pretty much as certain as you can know anything with genome sequencing. They're just too crazily different. That can't evolve. You can't get that kind of difference to evolve in 5, 6, 7,000 years. So it's a wild species. But this is not an uncommon problem in the annals of domesticated plants, that we don't know much about our wild ones.
Thank you. I think we are to the last question now. Dr. Sunil Mahajan is asking from India. Is there any Gossypium species which bears more than 5 locules in a bowl?
Regularly, no.
Okay.
No. Not one. Yeah.
Thank you so much. I think we are through with most questions. There is just one more. I'm sorry, Kai. This is just the last one. This is from some anonymous attendee. He calls himself anonymous. Are seeds of whole wild species set available to establish a complete herbarium of them?
No. I mean, I would love you. So, you know, I've been struggling to grow as much as I can in my Iowa greenhouse. Now, you know, Iowa is north temperate zone with terrible blizzards and winters. And I mean, as bad as, and no, if you've heard about how bad the climate is in the north central United States, it's actually much worse than anyone will tell you. It's terrible. And so I do my best, but I'm not even funded to do this. I don't have anybody working on the wild germplasm. I've just been doing it out of passion. And I try to send my seeds to the only place where there is actual support for it and way too little support, and that's the USDA Cotton Germplasm Bank in College Station, Texas. And it's terribly underfunded relative to all of our other major crops in the US. I can't speak for the whole world, but in terms of US investment in important commercial plants, cotton is number 1 on the list to me of underfunded germplasm resources. And because it takes so much effort to get these things to set seed. Each one is different. Each plant requires different handling.
Thank you, Professor Wendel. That was wonderful. Thank you so much.
Thank you.
Yes, thank you very much indeed, Professor Wendel. I hope that you can stick around for the rest of the webinar. Have a look through those questions, and if there's anything that we've missed, please feel free to add a comment in there. So we now go from one giant to another, and it gives me great pleasure to introduce Dr. Ed Barnes, who is no stranger to many of us on this webinar. As I mentioned earlier, Ed is the Senior Director of the Agricultural and Environmental Research Division at Cotton Incorporated in North Carolina. His topic today is increasing automation and robotic applications in cotton. So Ed, he is well known to us. He's an outstanding scientist who has been leading efforts to increase the automation of cotton production systems. And for the last 18 years, Ed has managed agricultural engineering related projects, including programs on precision farming, ginning, irrigation management, conservation tillage, and cotton harvest systems. He has also played a significant role in documenting cotton's progress in reducing its environmental footprint, while at the same time increasing productivity, and he currently serves on the Science Advisory Council of Field to Market, the Alliance for Sustainable Field to Market at the Alliance for Sustainable Agriculture. Ed has received his PhD in Biosystems Engineering from Oklahoma State University. He's a fellow of the American Society of Biological and Agricultural Engineers. He was honored with the National Cotton Ginner Association's Charles C. Owen Distinguished Service Award in 2019. And in 2013, he was awarded the National Conservation Systems Precision Agricultural Researcher of the Year Award. And in 2014, the ASABE Mayfield Cotton Engineer of the Year Award. So lots of awards there, Ed. We're delighted that you're here to and join us today, and we look forward to your presentation.
Thank you. Appreciate that, Kai. Can you guys hear me okay and see my screen?
We can.
All right, great. Well, I really enjoyed Dr. Wendel's presentation. You know, I'll build on what Keshav said. I'd say he's like, and this analogy is probably made before, he's like the Indiana Jones of cotton genetics, from what I can tell. So that was just really great. And it's nice to be paired with him because he's given a wonderful history of cotton and where it came from and the genetic progress that's being made. And so perhaps you could put my presentation in the context of where we may be headed. So looking at increasing the automation and robotic applications in cotton. Before I move on and talk about my co-authors, if you're not familiar with Cotton Incorporated, we are the research and promotion program in the U.S. funded by U.S. cotton producers and importers of cotton. We do everything from research to textile development to research and promotion. To promotion. So with that, I want to acknowledge that a lot of the ideas here I've benefited from working with some really wonderful people across the United States and here at Cotton Incorporated. You heard Dr. Wendel refer to some Cotton Incorporated funding. Dr. Don Jones leads our genetics and breeding efforts, and he is, I know, mentioned Dr. Wendel many times. In this presentation, I've benefited from input from several disciplines in our research division here, including Galen Morgan, our Vice President Kater Hake, our economist John Devine, entomologist Ryan Kurtz. And I won't read through all the other names, so we don't spend all the time just talking about who's contributed to this, but you can see several different institutions in the USDA ARS are all helping us think about how cotton could benefit from advances in automated technologies. One thing I won't talk about, and this is what Dr. Hake has encouraged me to think about, I'm an agricultural engineer, and so I attack things from an engineering perspective. Kater has said, well, if you engineers can't figure it out, maybe the geneticists can help. So he's engaged Dr. Brian Eyre at the University of North Texas. If you think about the history of particularly robotic harvest in some vegetable crops, the engineers couldn't quite get it figured out, so the geneticists stepped in, and so the agronomists stepped in, and made it easier for automated harvest to occur. So that could be a possibility for cotton. I want to acknowledge that, and but today we're going to be focusing on some of the potential engineering solutions. As we go through this presentation, I will say it's going to have a bias towards the first part, particularly towards mechanized production systems. But when we get to the harvest part, and that's why I'm going to kind of use that as a hook to encourage you to try to stick with me for a few minutes, I think the harvest, automated harvest for cotton would have applications for a lot of different production systems. And as will machine vision, as I hope to illustrate in this presentation. You know, I was lucky enough to, thanks to the World Cotton Research Conferences, got to travel to India and before and after the meeting, visit some Indian farmers. And, you know, this picture is from that visit. And, you know, the hand harvest is really, as we'll see, is there's some benefits to that. But some of the Indian farmers, even then, were saying that, you know, labor cost is becoming a concern. So that's something that may have broad applications. So we'll get to that here towards the end of the presentation. So I want to start out talking about why automation, and then I'd just like to walk us through the cotton production process from planting through ginning and look at what are some opportunities for automation. And how did we get to this discussion? You notice I'm using the word automation more. I think sometimes when I talk about robotics, people think, oh, that's, you know, science fiction, that's 50 years away. And in reality, it's not necessarily the case. I think some of these things are within 5 years. Some of these things are within 10 years. So these are not, you know, the year 3000 type ideas. And how did we get here? So on the left side of this diagram, I'm going to call research and technology innovations that are driving part of this automation. In cotton, we've done work on sensor-based management. A lot of people have been looking at how to adapt imagery from UAVs to high throughput phenotyping. And as we've done that, we realize machine vision can identify different parts of the plant. And there's a lot of advances being made in modeling and artificial intelligence. So while we have all these technologies converging that are increasing our ability to do things, on the right side are kind of the things that I see our producers facing. One thing, and I'll say more about this in a second, on herbicide failures. And there's very, you know, as chemistries run out, we're running out of ways to address weed control, and then labor shortages. And we're seeing this, I think, around the world, in the US and Australia, Brazil. That machine that's pictured there, we'll talk more about later, is now listing for $1 million. And the reason a lot of producers are willing to pay that much is it reduces labor requirements. And then finally, a lot of equipment now is equipped already with auto steer. So once the operator points it in the right direction, it drives itself. So we aren't that far from autonomous equipment, and we're already seeing that, as we'll see in some of these examples coming forward. I just want to call out herbicide resistance, because this is one that has me very concerned. And you know, there was a paper published in 2018 called Weed Management in 2050, Perspectives on the Future of Weed Science. And they talk about, that's where this chart came from. This blue line is the herbicide sites of action that have been developed. And you can kind of see those new actions really kind of terminate in the 1980s. And then the red line are the weeds that are becoming herbicide resistant. And we see those have continued to increase. So that is very concerning. So one of the things in this paper that the weed scientists actually concluded is that robotics may be one of the technologies to help address herbicide resistance. And I think I bring that up because I think, and we're already seeing this, that the first application of autonomous equipment on the farm is going to be associated with weed control, and that's already happening. To get us excited, and after Dr. Wendel's really lively performance, I'm going to cheat and use a video from the University of Sydney. They are really one of the leaders in agricultural robotics. Thanks to Alan Williams of the CRDC. When I was over in Australia, I got to actually visit the lab where they're doing some of this work. It was very impressive. So I just want to play this video. I think I have the sound enabled. And so this is just a quick minute to show some of the things that they're doing at the University of Sydney. And I'll just let this play, and you can see some of the words in the front, just showing some of the things they're doing. And this one right here, I find very exciting. there it ends, talking about labor shortages as one of the driving factors. There's a couple things in that video I just want to call out. One was the potential for solar power. That you saw that machine. When I was there, they were able to run that machine with 20 hours based on solar power. So that's exciting. That reduces the challenges we have of figuring out how to recharge batteries in the field. And then you saw a great example where they were weeding lettuce, and there's a company, Blue River here in the United States, that was acquired by John Deere, that has developed a similar system for cotton, where they use machine vision to precisely identify cotton plants. And then if it's not a cotton plant, they remove it. And what I liked with this video from the University of Sydney, you saw they were using a mechanical cultivator to remove those weeds that wasn't relying on herbicides. So that's why I think weeds and herbicide resistance are going to be one of the driving forces that get us into the position to have these things. And if we end up seeing autonomous equipment come to the farm, obviously the most benefit to the farmer, the most way to make this as economical as possible, as feasible as possible, is to use these machines for more than just weeds and more just harvest. So let's kind of think about, if we have autonomous equipment come to a cotton field, what could we do with it? So first of all, we'll start with pre-plant and planting. The things grayed out where I'll have slides to address after this one. But just to start off with soil sampling, in many parts of the world, we're looking at managing within field variation in soil type and fertility management programs that are at subfield level. And so those are based on soil sampling that historically have been by someone walking through the field with a GPS, collecting a soil sample. It's very time-consuming, labor-intensive, and expensive. And so there is currently a company in the United States now, RogoAg, you can see their website here, that have created an autonomous bobcat that's been adapted to autonomously travel through the field, collect soil samples, put those samples in a bag, label those bags, and record the GPS where that is located. And one of the things we're seeing across the board in agricultural robotics is these first commercial ventures are being done as services. And so this is the first example. They are not selling at this time, trying to sell you an automated bobcat. They are selling a soil sampling service where they take advantage of this equipment to provide you with soil samples for the farm. Another one I won't have a slide on, but it's a really important one, I think, for cotton. Obviously, many of us have encountered times where there's been, in the U.S., sometimes in West Texas, there'll be a hail event after we've planted a cotton and it's emerged, or we have non-ideal conditions of planting where we get skippy stands, and the farmer has to struggle to decide, do I till this all up and start over and replant? Do I give up? So what if we could use an autonomous equipment to go in and just fill the gaps so that the whole field didn't need to be planted? So I think that's something that robotics will bring as an option in the future. Another thing where, if we have robots on the farm, in many parts of the world, we're trying to improve soil health. Part of that can involve planting a cover crop. In cotton, one of the challenges for cover crops is we want to make sure they get big enough to provide protection to the soil, to build organic matter. So sometimes planting them early enough is a challenge. So this is obviously corn, but this is a robot that's being tested in corn, where they can plant the cover crop before the plant is harvested. Another one, and this is again a commercial entity, Australia, I have to admit, is one of the leaders in agricultural robotics, and this is Swarm Farm. They're a commercial company in Australia that provide, again, as a service, they will bring a collection of these autonomous sprayers to your farm, and they will put down a burn down application of herbicides. That light is showing this as a see and spray system. So not only is the system autonomous, it's somewhat intelligent, that it only sprays when there's something green present. So that is already out there and working. Another kind of commercial example on the planting side, and this may not be my first choice to launch a swarm farm, but it is really interesting. AGCO is a large global agricultural company. They have a division in Europe called Fent, and that company has talked about Xavier. That is their concept for swarm farming in the future, and it's really interesting. In this particular application, they're getting a bunch of little robots. They bring them to the field and let them go plant. One of the things I'm going to kind of harp early on about these small robots, and that's because you see at the top of that slide, they address safety. You know, it is a little bit scary to think if we get a 60,000 pound cotton harvester autonomous and something goes wrong, it could wreak a lot of havoc. But if we have a small autonomous robot like this that fits between the row and something goes wrong, it's just more of a nuisance. It's not a safety concern. So I think from a liability perspective, it will be easier to implement some of these robots, small robots or small autonomous vehicles in the near future as opposed to completely autonomous large equipment. So now let's move on from planting to the within season possibilities. I'll say more about weed control in future slides. This bottom left is a little robot that's commercially available from a company called ClearPath Robotics. The model is the Husky, and Dr. Joe Maha at Clemson has been doing some tests with this. I'll show some other examples in the future. But again, what I like about this platform, it's small. It's already got the tools to quickly make it autonomous. Dr. Maha had this running autonomously through between cotton rows in less than 6 months because a lot of the tools are already there. So obviously, if we have a small piece of equipment like this where we can put cameras on it, it can definitely provide information from disease and scouting, even look at fertility status. Even introduces the possibility of putting on some late season fungicides where we could spray from under the canopy, or late season nutrient applications where it may not be feasible to drive equipment through the field. This top left one is kind of a cute one. And some of you may have seen, I didn't put it in here, but there's a video that kind of went viral on YouTube about a mechanic or kind of a robotic wolf in Japan that's scaring away bears. This is the similar concept. There's actually a US patent on this. Where we could put, if we have, like for example, in the U.S. we have some trouble with deer coming into the field. We could put a fox on top of that little Husky robot and scare the deer away. So that's just one other kind of unusual thing that we might be able to do once we have this autonomous equipment. Definitely, it's unlikely that economics would justify any one of these applications. But if this is just one of a series of things these little robots could do, it makes economics more potentially feasible. And this is just another example of Dr. Maha's program in Clemson. Here you can see there's the little Husky robot. He's kind of using it like a utility tractor, and he's pulling a little disc with it. So the idea that it could cultivate in between the rows during the season. And this is just a view on a camera looking back from the robot, and the robot's view looking forward. So they're just, and this robot's already out there. It's not inexpensive. Keeping in mind, this is a development platform. It's meant to be flexible to allow different things to be added and taken from it. But we're talking over, around $25,000 for that platform right now. Another approach, this is a company, again, they're offering this as a commercial service in the US, Greenfield Robotics, where they have a little swarm fleet of mowers, basically. These are a lot like that Husky, but the idea is that they go between the rows during the season and they literally mow down the weeds. So that's an interesting service on another form of weed control. I want to show one more video. And this is a European company, again, Eco Robotics. And I just like their concept, so I'm going to let this play. Let you enjoy the music. again, I really like their concept. Some of that may be a little aspirational, but I do think this is, this could be our future, where we have a solar powered autonomous equipment. It doesn't, and it's using machine vision. So we're getting ready to transition into this discussion on machine vision. But you saw that this system has that camera. It identifies the weed, and then it goes down and spray it. I also have seen another version of this where they put a little tillage tine, so it's not relying on herbicide. And again, I'm not downplaying herbicides. I'm just talking about our concern about that there's going to be some weeds eventually that herbicides are ineffective against. So this could be a little tine on the end of that delta arm would provide us, a way to control those herbicide-resistant weeds as well. So let's transition. And so some of you have probably heard about machine vision at this point. There's a lot of applications. A lot of progress is being made in terms of developing autonomous cars, and there's been, and I'll show some examples, whether you know it or not, it's already being used on your iPhone to help you categorize your photo library. It's being used by Microsoft. I'll have an example of that later. One of the challenges for applying, these machine vision tools and artificial intelligence to agricultural systems is, of course, we have varying lighting, the varying lighting conditions. It could be cloudy, it could be sunny. We have different sun angles, we have different soil backgrounds, we have different crop production, crop growth stages. And so in order to train these systems, these deep learning systems, they need to be able to address all of those for it to become practical. And so that means we need these very large, maybe overwhelmingly large image libraries in order to account for all these situations. And so different people have come up with a solution to that, and that's by generating synthetic images. I'm going to use this example from a university in Rome. I won't embarrass myself by trying to pronounce its name, but this is something they've done for sugar beets. And so they've developed, basically a 3D simulation of a sugar beet plant. They can, see they can randomize it. They can change the soil background. And then they can generate a synthetic field that's shown here. And so now if we had a weed, we could put it in the synthetic field and train the learning algorithm as the sun moves across our synthetic field. And actually, we are working with Dr. Matthew Pelletier, who's with the USDA ARS in Lubbock, Texas, and he has already developed a synthetic cotton field. And so what he's done, very creative, took one of these 3D gaming engines, and so he's created a 3D environment. These pictures here are not actual cotton fields. They are a simulated cotton field that Dr. Pelletier has generated. And you can see these two different conditions, how he's able to change the lighting conditions, change the shadows. And so I think this is going to be really important to accelerating our ability to do machine vision work in cotton. Why am I excited about some of this? There's some near-term applications, and the hardware cost of this is not extreme. I'm going to show later a platform from the University of Georgia that has developed a way, autonomous system to remove cotton bolls. And this is the hardware they used. And what's exciting about it is they use ROS, which is a robotic operating system. It's an open source system. OpenCV stands for Open Computer Vision. So these are open source software tools, so that reduces the cost and greatly increases the ability to collaborate on these projects. The Jetson 2 is the kind of the computer brains behind this, and they were able to get enough horsepower to do the image processing for real-time control of a machine vision system for under $500. So that was encouraging. And then they've been using the Z camera that, if you notice, there's two lenses. This is a stereo pair. So not only does this take images of the field, it does it from a stereoscopic perspective, so you can actually measure distance. So you would get both distance and imagery from that. And that camera, again, is under $500. So for less than $1,000, they were able to develop a control system to harvest cotton bolls. And obviously this has other potential applications. There's a lot of work beginning to emerge of people publishing image data sets that can be used for training these systems in agriculture. This is a review paper that we sponsored with Dr. Sierra Young and Yuzhen Liu at NC State. Now Dr. Liu is at Mississippi State. And so there's one of the ones that came to mind, Deep Weeds, is out of Australia. And it was kind of interesting. Obviously, I'm not a weed scientist, but when I looked at all the weeds in that database, I realized I had not heard of any of them. And so definitely there's a need for local databases in all of this. And that's why I'm very excited. Dr. Cranthey informed me, and he's been talking to us about collaborating with us on these image libraries, and ICAC, he tells me, has already acquired 41,000 images for machine learning applications. And here you can see it's major and minor miscellaneous insects and diseases for India and Africa. And then one of the challenges, the time-consuming part about this, is kind of easy, somewhat easy to collect the image, but then for some of these machine algorithms, currently the best way to get them to be most effective is to draw a box around, say, the weed or insect of interest, so that the algorithm knows that's what it's supposed to look at. In the future, there may be some automated tools to help automate that process, but it will still require human oversight. So that's exciting. And then as we apply these databases with the synthetic library, I really think this machine vision is going to really take off for agriculture here in the very near future. Here's an example of a US company called FarmWave. Currently, they're mainly working in corn and soybeans. And I haven't independently verified this, but in their literature, they talk about they put a camera on the tractor so that every time it travels through the field, it collects imagery, and they don't ask the farmer to go back and look at all those images. Their algorithms actually scan that image in real time and map out, give points of concern if there's disease or insect pests that are occurring. They also have a corn kernel counter. We're also working with them to help us see if we can develop some research tools to quickly count cottonseed. Dr. Galen Morgan worked with them on some planting seed, and we're going to start on fuzzy seed this year. So those will be some near-term applications of machine vision, and I'll have some others in a second. I mentioned, this is actually, it turns out, been, and maybe some of you are very familiar with this, this has been a feature of Microsoft Office for about the last 2 or 3 years. And so I just happened to notice this the other day. I was creating a presentation to talk about cotton and water. I wanted to show a desert. So I got off my computer a picture I had from Phoenix, Arizona, in the desert. And look what, I noticed it put it generated a caption for it. And I read the caption, and you see this is the caption that Microsoft generated. This is a picture containing sky, outdoor, nature, and it's cloudy. That's a pretty accurate description of that picture. And so I thought, okay, well, what happens if I pull up a picture of cotton and look at that? I don't know why Microsoft decided to figure out what a picture of cotton was and train for that, but of a field of cotton, but it actually identified that. And they say it even tells you its confidence level. So that's just running in the background of Microsoft Office. That just shows you where this machine vision tools are going and how ubiquitous they're becoming. So now we've gone through the within season. We talked about machine vision. Let's talk about harvest, where I think we have a lot of potential for cotton. Just quickly, for mechanized systems, and we're looking at, one of the challenges, I think this is true around the world, that there's a lot of plastic blowing around, plastic garbage bags, plastic shopping bags. They can get into the field and they become a contaminant. We're doing some work with UAVs pictured here in the middle that could detect, could find the plastic bag, and I could even envision where it flies down and removes it, or we send a ground robot to retrieve it. Another thing with these cotton harvesters, I'm going to explain more detail later, but of course they create these round cylinder modules, and those have to be staged for transportation to the gen. And so this is certainly where we could, I think in the near term, be able to have an autonomous tractor that handles that staging part, where we don't have to devote a person to staging these modules. But let's look at what are our challenges for those of us in mechanized countries. These machines, as I mentioned earlier, are expensive. The newest model, they went ahead and broke the $1 million US dollar list price. We've talked about that we are seeing some challenges with this. This is plastic that's encasing the cotton. Keeping that out of the processing stream sometimes can be challenging. And again, if you're going to spend $1 million on a harvester, you really need a lot of land to justify that investment. We, in round numbers, about 2,000 acres or 800 hectares per machine to justify that cost. Even in the U.S., that is a challenge for some of our producers. They're having to go in together. And custom harvest is a little more challenging. We are seeing that does work in some parts of the country. For example, in Texas, where they start in the south and then progress to the north. But a lot of areas of our cotton belt, harvest comes at the same time, and it wouldn't be practical to have equipment moving across the country. And then another thing is these are very, as I mentioned, these are very heavy machines. We're worried about soil compaction. Currently in the U.S., a U.S. cotton producer, and I think this is true in probably Brazil and Australia as well. If you want to buy a cotton harvester, a new one is going to come from John Deere. That's the only company that's offering that I'm aware of. And then, of course, we spend $1 million on that machine. We use it for a couple months, and then it sits in the barn. So that's some challenges for cotton. And in order to keep cotton economical and cost competitive with polyester, we need to look at what can we do to reduce the cost of harvest. So why robotic harvest? Again, this is one of Dr. Maha's work with the Husky in Clemson. One of the things, if we did have a small robot that could go between the rows, we would again have the ability to do frequent harvest, as many of you do now with hand harvest. So of course, relative to machine harvest, that means we reduce the risk of yield loss because we get the cotton before, say, a hurricane comes in. And also we prevent quality loss that might happen when rainfall occurs. I'll have an example of that later. And it removes that scalability issue. We can go, ideally, maybe this could be a small, one of these machines would cover somewhere between 10 to 50 acres or 4 to 20 hectares. And again, it wouldn't just be a cotton harvesting autonomous piece of equipment. It could also do our weeds, weeding, pest scouting, spot spraying, replanting, all those other things we've talked about. So it becomes a multi-functional machine. So this is an example of work that Dr. Glenn Rains at the University of Georgia. He had a graduate student, Dr. Conde Fu, who is now graduated at NC State. And now Dr. Fu was a very talented student, but in less than a year and a half, he was able to take that machine. You see that's a high clearance, essentially a high clearance tractor that is being operated and controlled by that machine vision system I showed earlier. No one is driving the machine and no one is controlling that arm. It's all being done by the computer vision system. And you can see it sometimes has a little trouble. One of the challenges, this machine uses hydraulics, and it's using the machine position to get a long row position. And so sometimes he had to actually account for the temperature of the hydraulic fluid so he could precisely control that machine enough. And they've advanced beyond this kind of artificial situation. They have had some success in the field in partially defoliated cotton. And so this really encourages me that for a limited amount of time, a limited hardware investment, a student was able to do this in a short amount of time. That just shows where this technology currently is. We don't have all the problems solved. I just want to point out that their new, the next platform, they're going to go to a smaller platform. This is, again, a commercially available autonomous vehicle called a rabbit tractor. And so they're going to scale that down so that it fits between the rows as opposed to that big machine. It also makes it a little easier to have electronic stepper motors, so it'll be easier to control the machine along road direction. Another challenge for this is making sure we have a good end effector or a good way to remove cotton from the plant where there's branches and other things obscuring, the ability to reach that bowl. This is one example of end effector that's been developed. Hussein, who is one of Dr. Alex Thomason's students, they started this work at Texas A&M, and now they've moved to Mississippi State. But this is one example of where these pins are rotating and then when it comes in contact with the seed cotton, it's kind of, the pins pull it up between these arms, and then it can be removed by vacuum and from the plant. So this is looking very promising. They're looking at several different iterations of this as an end effector. I will note that I am obviously most familiar with what's going on in the United States. I know there's a lot of global interest in this. I've seen papers out of India and China looking at robotic harvest. I know in India, I don't know the current status of it, but there was Green Robotics looking at developing a machine for cotton harvest in India. So this is becoming more growing interest in how can we use autonomous tools for cotton harvest. I'm not going to bore you with these numbers. I'm an engineer. I like to talk numbers. I would just say there's a few things where we've run some equations. And, you know, if we just, I'm going to pull one scenario. If we had a autonomous machine that could go between 2 rows of cotton at 5 kilometers per hour, and we wanted to harvest a field 10 times between the time the bowl opened and the time we considered the harvest complete, that we would have to harvest 9 bowls a second. And that sounds like a lot, but I do believe with some multiple arms, we could do that in the future. And we would need about 45 machines to cover 800 hectares. And if you say someone negotiates the price of a current cotton harvester for 800 hectares down to 800,000, we need 45 units to cover that same area. You know, now we're already talking from, this is very, well, I'm going to talk about a better economic model later, but this is just a feasibility in my mind at $18,000 per unit. That's not impossible. That doesn't sound out of the realm of reason. So that's why this is something we're still giving thought to. Another thing we haven't fully developed is looking at, you know, how the materials handling will go on once the autonomous equipment has removed the cotton. We've given some work, Alex Thomason and Joe Maha have considered this. We're also working with another professor, Ajay Chandra at the Kansas State University, looking at, you know, how can we most effectively move cotton around. One of the reasons I'm not as concerned about this is a problem a lot of people are working on for different commodities and even for warehouses. So there's a lot of algorithms being developed to optimize material flow. And so I think cotton's not going to have to solve that one. Some other people are going to work on that. Also mentioned service models. And so again, all the current applications I'm seeing of autonomous equipment in agriculture right now is a service-based model. This is an example I just saw recently. There's a company called Burrow, and it doesn't look incredibly exciting, but they are actually, what they do is they provide autonomous carts to vineyards in California, and this eliminates the need so the workers can remove the grapes, and then the cart handles picking them up and taking them to where they go autonomously. So that's a service that's being provided. Just another example of one that's you know, going on right now. There's a company called Advanced Farm that is addressing strawberry harvest. So these are their strawberry harvesters. And I talked about, you know, one of the challenges for machine vision in agriculture is lighting. The way they've addressed that, they put big canopies over their equipment. And so if you look under that canopy, they have these mechanical arms that are going down and they're pulling up the strawberries and then placing them in a basket. This is all, again, happening under the canopy in the field. And then, you know, they're done. So this is, again, a commercial service that is being provided to strawberry producers. And so if we can do it in strawberries now, cotton is actually much easier, right? So with strawberries, if you think about it, number one, we need a gripper that can be gentle, that it doesn't squash the strawberry, and that it can, you know, gently pick it up and move it. We also have to make sure that strawberry is the right color red and that it's mature. And so with cotton, we don't have all those challenges, right? If it's white and it's open, it's ready to harvest. We don't have to be incredibly kind to it. You know, it can handle some abuse. It can be conveyed through suction, through air. And so in reality, cotton's an easier problem to solve than vegetables. And the vegetable guys have already obviously made tremendous progress. So is this our next cotton harvester? On the right, you'll see pictured, this is from last year, Agritechnica, John Deere showed one of their concepts for an autonomous sprayer. So on the left here is like, could we put this could either be an independent little robot that runs between the rows. And so this blue area, this is my concept of these would be the end effector that comes out. There's a detection system. It's looking forward, so it has time to think. And then tail, this would be a stacked array of arms. So maybe five little arms here that when the bull's detected, it deploys the arm and that end effector pulls the cotton back in. So that could, and you see the way this is arranged, you could envision this as an autonomous piece of equipment, or maybe an intelligent header that then goes on the sprayer, that replaces the spray boom, and now this autonomous sprayer becomes your cotton harvester. So I think there's a lot of possibilities here. As we finish up and start talking about cotton ginning, especially for mechanized systems, one of the potential benefits is we'll be more precisely removing the seed cotton, so there'll be less processing needed at the gin. And then we definitely, we did some studies where we did multiple hand harvests at different locations in the country in 2018 and 2019, led by Galen Morgan and James Griffin at the Texas A&M. We had studies in Georgia with John Snyder, and in West Texas with Amy Cura, and then Tyson Raper in Tennessee. So we went in and did frequent hand harvest. This is just one example for the site in Texas. You can, on color grade, the easiest one, you can see these are individual harvest events. So where they just went in and handpicked whatever bowls were open, obviously, as you'd expect, very good color grade. Then there was a time where it rained and they weren't able to get into the field. And we all saw, just from that one rain event, we saw a decline in the color. These last 2 points, this is where everything was hand harvested at the end of the year, and then the far right is machine harvested. So there's definitely some quality benefits. We also speculate there'll be increased fiber uniformity, and again, lower the risk of problems due to things like aphids and whiteflies causing stickiness. So when we look at ginning, there's already a lot of automation occurring in ginning. A lot of the gins here in the US, and I know in Australia as well, have automated strappers at the bell press. We're now seeing automated baggers and samplers. It used to be, not 5 years ago, there'll be 6 or 7 people standing around this bell press. Now there's often just one person. We haven't seen this in cotton yet, but the fact that there are autonomous forklifts has a lot of implications for both the ginning and warehousing of cotton. I see that coming in the future. And just this picture at the bottom, I mentioned the plastic, contamination problem. A group at the USDA Gin Lab in both New Mexico and Texas have developed a system that uses machine vision to detect plastic on the feeder apron of the gin stand, and then it sends a puff of air to remove that. And that's been picked up commercially by Lummus and Bratney. And so that is another example of machine vision being used today in cotton production. Not to say we won't have some challenges. This will have to be reliable and durable. I show a picture here of Dr. Bob Nichols. I just want to pay some homage to him. He was our agronomist for many years here at Cotton Incorporated, passed away last year due to ALS. And he used to challenge me. He said, "Ed, you got to get your engineers working on weed resistance. We're running out of options." And so he worked on the challenge of herbicide-resistant weeds a lot. And so we're going to have to face additional challenges if we try to figure out how to increase automation in cotton production. And you see even things like vandalism and theft. If the fields are spread out, do we transport equipment between fields, or do we assign one robot to a field? And then of course cost. And so we do have, we're working with an economist at Kansas State, Dr. Greg Abendal, has a very detailed model pictured on the top right, where we can put in rainfall probabilities, probability of hurricane, estimates of cotton loss, estimates of quality decline, to look at some of the value of frequent harvest and allow us to do a very rigorous comparison between a frequent harvest system, autonomous frequent harvest system, and once over system, and then Dr. Terry Griffin has worked kind of with, I'll call it an empirical model to look at days available for harvest and how, again, how multiple harvesters could be compared to a single once over harvest. Our intellectual property strategy on this is to go open source. Cotton Incorporated is not going to manufacture robots. What we want to do is lower the bar for companies that are going to bring agricultural robotics or autonomous equipment to agriculture. We want to make it as easy as possible for them to adapt that to cotton production. And the last thing I'll say about some of Microsoft stuff, these icons you see here, this was a bulleted list, and I asked Microsoft said, would you like to look at design ideas? And it pulled out all these icons. I did not select these icons. So I guess because I had the word harvest, it pulled up this kind of fall harvest basket. And then I talked about university. So I guess that's our university course, robotics, and then agriculture. So that was pretty smart of our Microsoft system to come up with that. There's a lot of resources out there. There's just a Council for Ag and Science Technology paper that came out this year, or sorry, at the end of November of 2020 that talks about opportunities in agriculture. There's a lot of resources I'd be glad to share with you. I know I'm running out of time. So with that, I'll leave you my email. And if you do have questions we don't have time to address, or you're looking for some links, you're welcome to send me an email. So with that, thank you, Kai and Keshav.
Thank you, Ed. Really enjoyed that. As you know, I love technology and innovation. So I was really, really pleased to see all the things that you're doing. And for those of you who are able to attend the plenary meeting in Brisbane last year, yes, 2019, we were treated to some of that technology, seeing some of that technology there. You very kindly mentioned our image library of 41,450 images, which is associated, I think actually, Keshav, it's the largest image library at the moment associated with cotton pests and diseases. And it's growing. We haven't just stopped there, that's growing. And so we're really proud of that. And we're also really proud of the work that we're doing with you, Ed, at Cotton Incorporated on our soil and plant health app. And there was one question from Deepak which asked, which is the best platform for image processing and deep learning for plant disease prediction? It's exactly that. It's our soil, plant and health app, which we're developing with Cotton Incorporated and Cater Haig. So we're very, very proud of that. You will know that we at ICAC have a major project using virtual reality, and that's the big challenge with all this technology for us. Is it's brilliant, but how do we then get that technology down to small farm holders in India and Africa? And we have been using virtual reality training, and we're rolling that out. And we're also looking at using HoloLens, the Microsoft HoloLens 2, which is augmented reality. So I'd be really interested, Ed, if Cotton Incorporated are doing anything around augmented reality for cotton production. You can use the HoloLens, and HoloLens has been used in the textile industry, but how do we get it down into cotton production? So there's a question from us for you, and then we'll start with the main questions.
Yeah, so yeah, in terms of augmented reality, I have heard of some people in extension, not specific to cotton, looking at that technology. I'm not aware of specific applications in cotton right now, but of course what I mentioned Dr. Pelletier do isn't really augmented reality, but I'm on a simulated reality. I think, again, there's going to be a lot of potential for that to accelerate our machine vision work.
Good. Okay. Well, if you come up with some ideas, give us a call. We're looking to roll out some of our ideas on augmented reality.
No, that sounds exciting. Yes.
Okay. Let's go to the questions. So don't forget, put the questions in the Q&A box, because we're not looking at the chat box. So if you put the questions in the chat box, transform it into the Q&A box. So we have one here from Tansuk Ok. how can the cost effectiveness of robot-based harvesting be gradually increased? How can its potential effects on employment be mitigated?
Now, that's an important question. And anytime, whether it's cotton or any application of autonomous systems, we worry about displacing human workers. In mechanized systems, I think our challenge would be to make sure we don't increase labor requirements. So, but when we get into looking at the hand harvest systems, that is a concern. And one of the things I hope will happen in the future is, and it's starting to happen now, as there's more economic opportunities for people, new industry, higher paying industries, that part of it will be the competition for labor. And so we won't be displacing workers. It will be a way for farmers to replace missing workers. If that doesn't occur, I think there was a good study done by the World Bank on the mechanization of cotton harvest in Uzbekistan that was published in 2016. And it addressed the social impact of mechanized harvesting there. And they did point out that there may be a need to create industry for displaced workers. So that is something we need to be concerned about and keep our eye on, and make sure that that we don't take jobs away without replacing them with something.
Great, thanks, Ed. A question from Mohammed Akhtar Ruzaman. Is there any sensor-based insecticide spraying drone technology? And is it possible to develop any small cotton harvesting robot which can operate within 30 to 40, he's put decimal, of land area? I'm not sure what that means.
It's just a small scale.
Thank you, Keshav.
Yeah.
Yeah, so, well, on the UAV, I do know here in the U.S., there's a USDA aerial application branch with USDA-ARS at College Station, and they have been looking at using UAVs to deploy insecticides and to deploy, any different types of pesticides. And so that technology is being developed. The regulatory side of that's a bit challenging right now in the U.S. That UAV and the person piloting that UAV or supervising that UAV has to essentially pass the same piloting requirement that an aerial applicator would. So the bar is pretty high, but definitely the technology is there. Another, with UAVs, the challenge is payload. So water is very heavy. So it would have to be, on the other hand, we can do very precise application. So I do see that as for some applications where we have spot spraying, that will definitely be something that will be occurring in the future. In terms of, I do think, for some of these small robots, looking at this, that it could get, that's what I like about them. They're scalable. So I do think we could get one, as small as maybe, that would be cost effective on a 5-hectare farm in the future. Right now, we're not there, but looking at how cost effective this technology could become, I think it really may have a place in smallholder farms.
Thanks, Ed. A question from an anonymous attendee. Not sure how we have anonymous attendees, but anyway, the harvesting machines are very costly. Is there any subsidy given by the government of the USA to purchase such costly machines, or can we hire them?
Right. So there is no, cotton farmers would love with someone to help them pay for the machine, but it's actually that machine cost is like I said, is a challenge here in the United States. What we've seen is sometimes neighbors getting together where maybe they're not growing 2,000 acres of cotton, but combined, or maybe 2 or 3 of them going in and purchasing a machine. And so again, I think this is an economics problem, and we need, I believe, globally, we need an alternative harvest system to keep this cost down.
Okay, thank you. I'm just looking at one from Dr. Sabesh. Keshav, can you help me out on that?
Yeah, thank you, Kai. Actually,
try and make that into other English, I suppose.
Yes. like this concern being expressed by Dr. Sabesh is that with the use of heavy machinery like tractors, there is a problem of soil compaction. It is not only in India, it's also true with many other countries like Australia. So he's asking if it is possible to cope up with the automation by tractor-drawn implements. Is there something that can prevent this compaction with automation?
Yeah, and I do think, you know, the biggest answer there is the small equipment. Is, you know, for example, that ClearPath Husky, I think is under 200 pounds, and you know, not only does it reduce the potential for compaction, it also increases probably the time that you could get into the field, because it is light equipment. It could definitely get into the field under wetter conditions than a big machine could.
Good. Thank you. Thank you. I think that is all the questions. Well, if anyone thinks of anything, you've got Ed's email address. Just drop him a line and I'm sure he'll be delighted to reply to you. So that is it for today. I promised you a treat and we were given exactly that. Two giants in cotton research gave us fantastic presentations, and I'm deeply grateful to you, Ed, and to Jonathan for sparing the time to be with us and share your knowledge with us. I say it every time, I learn so much from these webinars, which I suppose coming from almost nothing is quite easy, but I'm sure a lot of you are also learning a lot as well. So that is it for today. The next seminar, Keshav, perhaps you could let everyone know who's going to be speaking on that and when it is. So from me, thank you to everyone for attending. Thanks to our speakers, Professor Wendel and Dr. Ed Barnes. Keshav, I'll leave you to just tell everyone about the next seminar.
Yeah, thank you very much, Kai. I'll be sending a mail with all the details. So I'd only like to thank Professor Wendel and Dr. Ed Barnes. Thank you so much. Thanks.